Scientists studying transcription yeast created an experimental strain that produced a modified RNA polymerase containing a single amino acid substitution. Okay, and the scientists were able to find the maximum elongation rate with and without the modified RNA polymerase enzyme. Okay, so I think we can tell from the first paragraph what they're testing. So we can now tell what's the dependent variable and also what's the independent variable, right? Can someone tell us what is our dependent variable?
Elongation rate.
Yes,
So
because
what does that mean?
that's what they're finding. They're finding it, they're determining it. It's called the maximum elongation rate. Okay, with and without the modified RNA polymerase. That means your independent variable is going to be either having the RNA polymerase or not having it. That's for the first paragraph. Now, there's a compound called Amanitin, which is found in toxic mushrooms, which is an RNA polymerase inhibitor. So it will bind to the active site of the RNA polymerase and it's going to inhibit what it does. It's going to inhibit transcription. In a second experiment, which is the one in figure 2, they treated the wild type and the experimental type with a 40 microgram per ml of amanitin, and they recorded the elongation rate. Good? Okay. Yeah. So do we have any amanitin in here? No. And what?
Can you explain the question?
Yeah. Well, the question. You mean the formation of the question?
Yeah.
Okay. So let's just go back for a second. Look, the scientists are doing what? They are studying the.
Amino acid, something RNA.
They're studying the modified RNA polymerase in a strain that's called the experimental strain. Okay, that's number one. Number two, how is it modified? It has a single amino acid substitution. Okay, how would they know the ability of the working of the RNA polymerase?
So I have a question. Isn't single amino acid substitution deletion or?
No, it's mutation. It's not deletion though.
It's not frame shift?
No. No, that's if you delete or insert. If you substitute, is not.
What is it then?
It's called missense.
Okay.
Yeah.
Do we need to know that?
Yes, you do. Okay. Now, they will check the maximum elongation rate with and without the modified RNA polymerase. Is the thingy dim? Should I increase the brightness?
Yeah.
It's a bit dim. Okay, I think from the back.
So you said increase, but you decreased it.
Yeah, it turns out it's not from me. It's more from them. Oh. Okay. Is that 100% brightness? Good?
Yeah.
Okay. All right. Oh, shoot. Okay, good. Now, there's a compound called amanitin. Do we know what it does? What does it do? It will inhibit the RNA polymerase. Good so far? The amanitin will bind to the RNA polymerase active site and is going to block the function of this enzyme. Now, in a second experiment, in a different experiment, they took the wild type, they took the new strain, and they gave both of them amanitin, and they recorded the elongation rate of the mRNA. Okay? Yes? We good? Okay. So here it's normal. Here there is amanitin. Okay, part A. Describe the three structural components of an RNA nucleotide monomer. Can someone tell us what those are?
Me. Ribose.
Ribose sugar.
Phosphate group.
Phosphate group.
Nitrogenous base.
It's not enough.
What-
Elaborate on the nitrogenous base.
ACGT.
It doesn't have T.
A U line, it's hard, I forgot.
So it's going to be A,
CGU.
C, G, U. That's how you answer part A. Now the second part of part A says explain the role of RNA polymerase during transcription.
It starts the.
They add the nucleotides?
Yes, it adds mRNA nucleotides or RNA nucleotides.
Adds RNA nucleotides.
Yep, to DNA template.
Mister, doesn't it just start the transcription?
Well, they already said transcription. So,
And what about the promoter region?
what about it?
How it binds to it? Start the transcription.
That's not the job of it. You could say that, but that's not the job. Okay? To make a complementary mRNA sequence. Good? That's how you answer part A. Okay. Now, let's move on to part B. Part B says, identify the dependent variable in the experiments. Which means you should look where?
Elongation rate.
The y-axis, the maximum
Elongation rate.
elongation rate. Okay. Identify a control group missing from the second experiment.
A group that doesn't contain that's Amanitin solution.
Exactly. Look, they said here in the second experiment, the scientists treated the wild type and the experimental strain with Amanitin. But how about without Amanitin? Right? They didn't do that. So basically, finding the elongation rate.
Isn't it the one right beside it though?
It is, but they said second experiment. Okay, that's
So
why.
I don't get it.
Okay, so look, in the second experiment, which is this one, imagine if the one on the left didn't even exist. Just imagine, okay?
So we're talking about the experimental strain.
Both the wild and the experimental strain.
What is
Look.
it missing?
The second one, the normal natural one.
Look at the caption of figure 2. It says we have Amanitin, right? But do we know the impact of not having Amanitin in the second experiment? Is it mentioned? It's not mentioned. That's called your control. How would you know if this is low or high? You must compare it to what? There's no control. Okay. And the wild strain and the experimental strain without Amanitin. Okay. Justify the need for this control in the second experiment. Why do you think control is important?
Show how the solution
Compare it
affects.
with other.
Right, so to see the impact of the Amanitin, to assess the impact of the Amanitin on the elongation rate. Because right now, they both look low, but are they truly low?
No.
Where's okay, where is our control? We don't have our control, right? To assess the impact of Amanitin, okay, on the elongation rate. Okay. Part C. Describe the effect of Amanitin on the maximum elongation rate for the wild type and the modified RNA polymerase. Now, what shall you do? You must now compare the wild type on the right to the wild type on the left. You must compare the experimental on the right to the experimental on the left. Okay? So comparing them means comparing their error bars. Now looking at wild type on the left versus the right, Did the Amanitin decrease?
Yes.
Yeah.
It did, right? Because if you guys draw a line from here to there, it's, you know, very low compared to the lowest error bar. So we can now confidently say that Amanitin decreased the maximum elongation rate in the wild type. But did it change anything about the experimental type? Let's check.
Oh, I didn't.
It did not, because right now if you draw an arrow from here all the way to here, are they overlapping?
Yeah.
Yes, but no effect on the experimental.
So it still decreased. So how can you say no effect?
You must compare experimental and the Amanitin versus without it.
So if they're overlapping, even if there's a difference, you're still going to see no effect.
Exactly correct. Yep. Okay. So determine the ratio of the average maximum elongation rate for the modified RNA polymerase compared the wild strain in figure 1. The modified is which one? The experimental strain, correct? How much is the y-axis here? How much is this point?
3,
Is it 3?
2 or 3.
It is, I think, 2. Yeah, it's probably 2. Draw a straight line from here to here. That's at maximum at 2. And how about this one?
12.
12. They're saying experimental to wild type. That's going to be
2 to 12.
2 to 12.
1 to 6.
Or 1 to 6 is up to you. Okay.
Yes, sir. This is where the question starts to not make sense.
Part D?
Yeah.
So A through C made sense?
Yeah.
Well, good. Okay, part D. State the null hypothesis for this experiment in figure 1. What does null hypothesis mean?
We studied it, Mr.
Right.
Was it in Unit 7? No, it was in Unit 6.
Okay. What did we say it is?
No, Mr. My point is we did study it, right?
Right, what is it? What is it? Can you
No,
say
I
it?
forgot.
Okay, so guys.
Mr. I haven't reached that part of the recording yet.
Oh my god. No more recordings, only the blueprint. Do you guys know what that is yet?
No.
I don't know.
What it means?
So wait. I have a
What it
question.
is? No.
If I start studying right now, what should I study?
The blueprint.
Okay, what else? Like, I want to fully start studying right now.
The blueprint.
Mr.
Just
what
that?
is the
Just that.
Nothing else?
What is it?
You need to study Princeton?
No, just the blueprint.
So where can I find it?
And the main page. Blueprint.
But Mr. You're saying if we study the whole thing, we can get a 5 out of 5.
Yeah.
If we're like,
Yeah.
even if we don't know anything.
Yeah.
Do they bring past like questions from?
The ideas will repeat the
So I
ideas.
study all of these 3?
Look, if you click on that one,
Yeah.
it tells you what files to go to. And tells you how many you have to do. And if you click on this one, it tells you, you know, the files that you should go to.
Oh, there it is, blueprint.
Yep. And to keep track of what you're doing, click on this one and just tick it when you finish it. So you do a file ticket. Tick,
Mr. How
tick,
long
tick.
would this take?
From now till the test day, you must get them all done. As long as you avoid everything else.
So for one day we can do like one.
No, it's not enough. I think you must do 2 to 3 a day because you guys started very late with the blueprint.
So I have another question.
If I start next month, no, start today, man. Please, start today.
So if I start studying for SAT right now, like for tomorrow's SAT, what should I study?
I think you should just give it up. No, go to Blue Book.
I have your SAT gen thing.
Okay. Go to the Blue Book though. The Blue Book, right?
So I've done every
Yeah.
single practice on Blue Book.
Even the new ones, they posted like 2 new ones.
Three new ones they posted out, did it?
And you did them all?
Yeah.
I
Okay,
need
then.
to check if the blue
should be
is
fine.
right there.
Okay.
Okay. Part D. State the null hypothesis for the experiment in figure 1. Guys, In figure 1, we are looking at the maximum elongation rate in wild strain versus what? Versus experimental strain. If you guys go back to the main paragraph, the first one, it talks about the maximum elongation rate due to the influence of the modified RNA polymerase.
So the maximum elongation rate does not affect whether having or not having the RNA polymerase?
Exactly correct, the modified one.
Mister, I have a question.
Mm.
Why does the test of 2020 have so many parts?
It's always has been like that.
Why? Was it because of COVID?
Oh, you mean like the, yeah, because of COVID.
They
It
had
was,
3 different tests?
yeah, because it was online at the time.
Oh my god.
Alright, guys.
Also, in your jacket.
So the maximum.
4 hours?
Yeah.
I don't know.
What is this?
Alright, guys. The maximum. Yes.
Can I show you something?
Yeah.
I'll show you this. Mister, look. Look at this.
No, Mr. Don't look.
Read.
Well.
So I was behind the scenes.
We start running because we're scared.
Well,
You guys want to see the TikTok?
guys,
Turn it.
let them see so they can know that what's going
Did
on.
it in school?
Yeah.
Wait, was I the cameraman for this one or was it
No,
the
it's
camera?
bad.
No, I'm not chasing after this.
So,
Thank
Mister,
you.
I solved the blueprint instead of doing these.
What is that file?
Practice part 4, 3, 2, 1.
They are. This is the proof.
No, this is daily practice. Leave that. Leave that. Just do the blueprint.
Mister, what do you mean daily practice? Like, do you
Supposed to
put
practice daily.
Guys. You want to get a 5 or what?
your 5, Mister 5?
That's it. If you start today, please start today, okay?
Mister, I have been started.
With the recordings, I want the blueprint. The
Mister,
blueprint.
you're not even posting the recordings. I have to search like.
Yeah, because you should not even do them right now. Right now is just time for the blueprint.
Mister, I need the recordings to solve.
The handwriting.
So I'll solve tomorrow after my SAT.
Okay. Guys, the blueprint from now till test day, you should get that 5. Okay. Print the to-do list and tick it when you get done with the file. Alright. Maximum elongation rate isn't affected by modified RNA polymerase. So what's null hypothesis? You simply go against what the researchers think. So if the researchers think that elongation rate will change, you just say it's not going to change. Okay? Always saying no change. Provide reasoning to justify the claim that the change in the amino acid sequence in the modified RNA polymerase affects the shape of the active site. Well, basically, a change in amino acid sequence changes the tertiary structure.
Only?
Not only, but this is the most important one for functionality wise.
Mister, is this question considered hard?
No, like medium, I would say.
This is a hard.
Medium. And the structure determines function. Let's see if that's what they wrote. Does anyone have the answer key open?
Guys, should I print you guys the to-do list thing?
Yes.
Yeah.
Okay, let me find my print.
Do it right now.
Well, it would be good if you could actually print it for them
There's
because
a printer right here.
I don't want them to go back to anything but that. They have no excuse, okay?
Mister,
Yes.
do you make the practice tests?
No, I don't make anything. I just
College course.
very heavy printer, man.
What is about? What if you didn't study anything?
It doesn't matter. It doesn't matter. This is regardless of your background. I swear.
Don't print one for me. I
So
have
now
one.
it's saying 2029 frequency. What does that mean?
Frequency. Mister.
Yeah.
No features.
Sorry?
Mister Miriam was like, it says 2019 frequency instead of FRQ. Girl, it's FRQ. She thought it said frequency. She's like, what's frequency?
Mira, shut. Listen.
I think we are
No, it says 2019 higher.
not
FRQ.
qualified
We're
for the
not
blueprint. No, we are qualified, guys. Please. Anyone can do the blueprint. Anyone.
Why is yours like this?
Look what.
Okay.
Oh,
If I
So,
get
yeah.
everything and still did not get a 5, what happens?
Then, well, look, like,
Actually?
yeah.
I think you just have to improve over time. Like, you cannot, let's say, get a 1 the first test and then get a 1 the last test. You know what I mean? That means you're not even doing anything. So, but you know how you should stay like some files to get a
I'll
5?
get a 6 for you.
Please, just like take it seriously, man.
Mister, I have been taking it seriously. Do you not believe me?
You're taking the recording seriously.
So, Mr.
So
we
I-
don't study the Princeton, nothing, only the blueprint.
Only, please, only the blueprint.
Only the blueprint.
Okay. So, anyways, so the maximum elongation rate is affected. Because if you change the active site, you're gonna affect everything else.
Mister, can I show you something? Look, I have this in my wallpaper. It says we have.
Oh, the
15
countdown?
Yeah.
59
Oh my God,
days.
that's scary.
Yeah,
Why are
that
you
sounds
doing
like I don't know nothing.
Girl.
Alright, so let's go ahead.
For instance, 59 days enough for the
Doesn't
blueprint.
this stress you out?
Like, how much are we supposed to study a day?
3 hours.
How many files are there?
You just
بس
said
progress
41.
check سهل ترى.
41 for one picture. There was like, I think 24 more or something.
More?
What?
There's like 65
65.
files.
Wow.
Girl.
What if
Wow.
we're not prepared and like we didn't study Princeton? Right?
It doesn't matter. You will catch up as you go on. Don't count Thursdays. Don't count Eid. Let's.
Okay.
You guys study on Thursdays?
Yeah.
Mm-hmm.
You actually do?
I actually studied like 3 hours yesterday. What?
Really?
Yeah.
Okay. Then count them.
Mr. I study on Thursdays, but I have to.
I thought people don't study on Thursdays. I don't know. Felt wrong, I guess. All right, guys. Question 2. Scientists studied soybean plants grown in sunlit controlled environment chambers with carbon dioxide at 700 micromole of CO2. Anyways, now the CO2 concentration in the chamber was double that of the normal environment. Okay, how do we measure photosynthetic rate? By knowing how much CO2 was exchanged in an area of the leaf. Okay, because the rate of photosynthesis depends ultimately on the plant enzyme rubisco. And during photosynthesis, what does rubisco do?
It's in the second stage.
Yes, it will fix the carbon into a bigger molecule in the Calvin cycle. Now here we have multiple temperatures from 25 to 50. What do you guys think is the best graph to represent this data?
bar graph.
A bar graph or a
Yeah.
line graph?
Bar.
Bar.
Because it's not categorical.
It's actually,
bar graph.
I'm sure they're both accepted, but line graph is better.
Mister, we haven't done a line graph till now.
We will, if this one requires drawing. Okay. Yeah, it says line graph actually for this one. Because temperature is like time. And time means line graph. Okay? So, describe the effect on an enzyme of an increase in environmental temperature above the optimum temperature. Guys, if there is more temperature than the enzyme can take, what happens to the enzyme's ability to function?
Denature.
Right. So the enzyme will denature and lose its ability to be functional. So the reaction rate is reduced. That's part A. Part B. Using the template in the space provided for the response, construct a graph based on the data in table 1. Okay, now let's do the plus and the minus thingy that we have discussed before. So like
100.
90, okay, that's 100.
82,
82.
101, 91.
101, 91.
101,
101.
1
101.
85.
85.
Mr. What's this?
We're doing plus and minus by the 8,
No, I don't
by the
know.
7.
I'm saying it like that. I don't
We're
know.
doing plus and minus.
Oh, okay.
111, 96, right?
7.
7,
Yeah.
97, 78, and 64,
64.
63, and 55. Now let's take a picture of that, and we're going to go ahead and
Take a picture.
we're gonna plot it. If you guys, how about you print us a template if you can, so they
I don't even
can.
know how to print this thing. I'm waiting for my
This
dad
thing.
to print it.
Oh, okay.
Do you know how to print, Mr.?
Monsieur,
I think
if you print it, can we reuse it for the same questions or what?
the template I have it. No, some questions have different templates. Like for
Oh, yeah,
example,
the different square numbers.
yes, exactly.
Okay, so if I like take a picture of this and print it, how does it work?
I don't know how to print. Is it Bluetooth?
No. Yeah, you connect with like internet.
Really?
Let me see.
Shall I?
I'll send it to you.
I don't know, honestly.
It should you leave it on the.
Alright, so if you want, leave it for now.
That of top.
Alright, guys, so what's our x axis? Temperature.
Y-axis is the inside.
The carbon fixation.
Mr. Why is the temperature down?
Because we're controlling it.
Didn't you say it's like time?
Yeah, and time is always done. Carbon fixation.
Okay.
Okay, U mole over meters squared over second. Now, our temperature will range from 25 to 50, right? So,
Gonna skip count by.
yes, I'll skip count. Now I'm gonna put that 25. Then I think I will go by fives. I can afford fives. So each 2 could be a 5. Let's check 30, 35. No. Let's just see. Each, maybe 3 is a 5. So 30, then 3 again, 35, then 3 again, 40, 3 again, 45, and 3 again, 50. Yeah, because you guys have to cover 2/3 of the figure.
So can you write like with the pencil in the example graphs?
Yeah,
And you can go over.
but the template could be very thick like this, like the lines. So if it's pencil, it's not so clear. It's best to use a pen.
Yeah, that's
Is it a digital?
No.
No, FRQ isn't. It's hybrid.
Yeah, the FRQ answering sheet is on paper.
بس اعتقد هاي السنة تقدر تجاوب بالبنسلز حتى البقية من
Yeah, all of them.
Mr. Isn't it they don't count it if you write in pencil? I heard something like that.
That's before, now it has changed. Now it's pencil allowed. Alright guys, now look at your least data in the carbon fixation. 55 up until 111, correct? Let's do a double tick. Maybe I can start with 50, right? And let's say each tick is a 5. Let's see if that will do. 55, 60, 70, 80, 90, 100, 110. No, I failed to cover 2/3. So we must start with, let's say, 40, then 50, 60, 70, 80, 90, 100, and 110, and 120. Okay,
Who's fasting you?
Me.
Okay, so basically everyone.
so 25 is the temperature. It's fine, I'm good. I'm good with that. Okay, 25 is the temperature. Guys, look at the first row. It's 91, right? So go all the way to 91, which is like here. And then you should do the plus and the minus, because we have to go up to 100 and as low as 82. That's the first point. The second point, 30 with 96, which is all the way here.
Monsieur, can you screenshot this after you're done?
Yeah. And plus minus 5, so 91 and also 101. Okay. 35 with 93, which is all the way here. And then 85 and 101. Okay. Now we're gonna have 40 with 104.
Monsieur, if I do intense like practice, how much will I finish? Miriam is saying she can finish it in 1 week. How much do you think I can?
1 week.
1 week is how is that possible again?
How is that
Study
possible?
like what?
We actually have a long holiday coming up,
One.
so maybe that's what she means. Is that what you mean, Mariam?
Yeah, that's what I mean.
45, 71, which is here.
So you mean a bit too much.
How many? Oh. 64 and 78.
He said yes.
Finally, we have 50 on the x-axis, but 59 on the y-axis. 55 and 63.
This is a line graph.
Yeah, but now we actually have to connect them by a line, by a straight line like this. 1, 2, 3.
So it's a negative correlation.
It has decreased. Yeah, it has decreased. You're right. Good. That's gonna be our line graph.
Monsieur, I have another question. Why does every test have like 2 parts? Like, no, I already asked that question. Like FRQ and then full test. Why don't we just do the full test?
They're both required because they're different from each other.
Why are they different?
The college board posts different things. That's why.
Just do it.
It's more practice for you. It's better. All right, now they're saying determine if there is a statistical difference in the amount of the CO2 fixed between 30 and 40. So are they overlapping between 30 and 40 Celsius? Let's take a look at it. Between 30 and 40. Are they overlapping?
Yes.
No.
Yes, they are.
40?
How?
Yeah, because look, draw a line and again hit that line. That's called an overlap.
So overlap is when the line hits the other one.
Exactly. Which means they are not different from each other. Okay? So if they overlap, they're not different from each other. And if you want to confirm it further, look here at 30 and 40 and see if the numbers overlap.
Yeah, they do.
They do, because you're going to have 91 to 101. You're going to have 111 with 90. We said 7, right? And they're overlapping in their numbers, right? Because there's 101, there's 97. So there's no difference, we would say. Not statistically different.
So I have a question. How many of these things do I have to do daily to be able to like have?
Well, count how many we have in total.
Yeah.
And like, be realistic with yourself. Like, you cannot study 7 days a week, right? Choose your maximum rate of studying that you've been doing. I think we had 65 files in total. Let's just say 65. And you have how many days left? 59 days.
60 basically. Yeah.
Don't count today, right? Because, you know, it's almost over. Don't count, let's say, some days of Eid, whatever it is. Don't count days you don't study on. How many days are left?
Let's say like 50.
Let's say 50.
Yeah.
So about 2 files a day.
Mister, every time I miss a day, I feel like I'm so behind.
Hey,
So
do the
probably
ticking.
the only day I'm gonna be missing is Easter.
Okay, that's good. You still have enough days. Just start, please. Start today.
Mister, I'll start on Sunday because tomorrow I have to study for 2 quizzes I have in school.
Well, here we go. We just lost 2 days.
Mister, you know I've been ignoring all my quizzes and studying only AP Bio.
Okay. I want to give that 5 then.
Do you support that?
I do support that. Alright guys, based on the data, identify the temperature range at which the soybean leaves are producing the lowest amount of carbohydrate. Well, they produce the lowest carbohydrates. They are what, here, right? Or here. So you can say 45 Celsius to 50 Celsius.
Decreased.
Yeah. And if you have less carbon fixation, that means they're producing less carbohydrates. Because carbon fixation just means producing carbohydrates. So we're going to say 45 to 50 Celsius. Okay, D. Increased amount of carbon dioxide in the atmosphere are correlated with an increased global surface temperature. Based on the data, predict how a surface temperature that continues to rise above 40 Celsius most likely affects the amount of energy available to primary consumers. Guys, if the producers are going to have less carbon fixation at high temperatures, you know what that means? They're going to make us less food. And if they make us less food, we will have less energy to consume, correct? So the prediction is that you're going to have less energy available. Now put a dot, justify the prediction. At higher temperatures, Robisco functions less, so less carbon fixation, so less carbohydrates produced or made by the plants, so less food for consumers, for primary consumers. Okay, one model predicts that an increase in greenhouse gases will lead to a 2% increase in average surface temperature on Earth. Based on the data from the experiment and the prediction of an increase in average surface temperature by the model, predict how the location of the plant species are expected to change over time. They want to avoid the high temperatures. They have to migrate to colder temperatures. They will migrate to colder temperatures. Okay.
So this is what happens when it decreases?
No, the temperature is becoming higher off the globe. So to avoid the high temperature, they go to an area of low temperature, which is the colder area. They immigrate, yeah, or migrate. Okay, very good. Now, so we have something called pigeon lice, which is a parasite that feeds on the feathers of the rock pigeon. The lice are host specific, and transmission of them between pigeons requires direct contact between the pigeons. Pigeons remove the lice with their beaks as they clean their feathers. And as you guys can see, you could have both types of lice on a pigeon. But I think it's pretty obvious that a white pigeon is going to have a hard time seeing a light louse, but it's going to have an easy time seeing a dark louse. And a black pigeon is going to have an easy time seeing a light louse, but a hard time seeing a dark louse. I think that's pretty obvious, right? Okay. Now, the hypothesis is that the evolution of the coloration in lice is driven by birds, by natural selection, basically. So they would take 25 lice from random sample of lice, okay, and half of the pigeons were fitted with harmless mouthpiece that stopped the beaks from entirely closing. The mouthpiece would prevent the pigeons from taking the lice out, okay, but they could still have food, they could still eat. Each pigeon was isolated and kept in identical environment conditions for temperature, humidity, and amount of light. And the lice will reproduce every 24 days. The researchers observed the phenotypes of lice over the period of 4 years. Describe the importance of phenotypic variation and louse body coloration among individuals. What's the importance of phenotypic variation?
Genetic variation.
Like the ratio of how many there are.
I don't think that's the answer. No, probably not. Okay. So due to phenotypic variation, some lice have more chance of surviving
How?
if their color matches the bird's feathers color. Yes, the bird's feathers.
What do you mean?
Well, like for example, if the bird is white,
You won't see it. The lice is white.
yeah, it can't see the white. That's what I mean.
But so I feel like if it's white, if you're in the dark, you can't see the white still.
Sorry?
In the dark, you can't see nothing.
Yeah, she's right. In the dark, you can't see anything.
So I feel like being white is better.
Don't think.
You know it's recording, right? So whatever you say is gonna stick. Now look,
So no one's gonna watch this recording.
how do you know?
Are you guys gonna watch it?
It's in the blueprint. That means everyone who knows how to get a 5 will have to go through this video probably.
Hi guys.
Part B.
But Mr. You don't mark, you know, Mr. One thing I found very confusing. You don't mark the recordings of the practice. Like you don't say, oh, we're doing practice 2020 in this recording.
Oh, I put it next to the file.
Meaning?
For example, right now, if you go to, let's say, test 2020, which we're doing right now, right?
Yeah.
I put MCQ solved right next to it.
So solved means it's the recording, right?
Yeah, from question 1 to question 60.
Okay.
I'll do the same for this recording, for the FRQ. So
Mr. You're gonna put
I did the
our-
same for, by the way, for test 2. I put the same. Okay. Yeah, I'll put this recording, the specific one.
Hi everybody.
Yeah, that's if they listen. Part B. Justify the decision of the researchers to hold lighting conditions constant in their experiment. What's the benefit of making the light conditions constant?
Like, what does that mean? What do you mean?
That means they keep the light conditions constant for both dark pigeons and light pigeons.
Well, the ones that match the skin color just don't die.
That's not why. That's basically
To control the amount of lice that are getting eliminated.
without light affecting. So basically to exclude light as a factor
Okay.
that may have influenced.
This question is not easy at all.
It's very diverse
I
in
found
its
this
answers.
question easier than the other
Yes.
ones.
بس فرنا 4 وايز.
Did you grade yourself?
When? What?
Did you grade yourself?
Yeah.
How much you get in this question?
What do you mean?
Like, did you check your answers with the answer key?
I didn't solve this question.
Oh, okay. Okay, now.
Find it easy.
I found it easier because you're solving it right now.
Predict the most likely effect of the pigeons cleaning their feathers on the phenotype of the lice after 4 years. That means, like, let's say over 4 years, the lice that have survived on the dark pigeon have what color?
Light. No, dark.
Dark,
no, because you can't
exactly.
see them.
Exactly. So basically,
I'm excited.
light lice being more on light feathered pigeons.
So can't you just make it simple and be like the one that matches their color, that's the one that
It's
survives?
easier than the other.
This is too complex.
Mister,
And-
are you checking the answer key?
Yeah, it's right there, and mine is as complicated as theirs, so we're good. And dark lice.
So let's say for that question, if I ever said the light lice goes with the white pigeon and the black one the dark one goes with the dark pigeon.
Yeah, that's perfect. That's what I'm saying.
But you say it in a complex.
Being more on dark, I am, but you know.
Mister, do we have to be like good with our words? Like, why can't
No, just communicate the idea.
Yeah.
So they won't detect marks.
I have to see how you write. I haven't seen that till now,
Okay.
so I don't know.
Keywords too.
The keywords, thank you. You should hit the
I'll
keywords.
start solving these and every time I come I'll show you.
I like it. I like it.
He
Okay.
does.
I think
Part, part D, provide reasoning to support your prediction in part C. Well, the lice with similar color
It's hidden. It's camouflage.
as the pigeon, exactly, camouflages,
blends in with this.
exactly, so survives and reproduces.
Mister, we spoke about lice so much, I'm starting to itch my head.
Okay,
Also, can you go back?
yes.
Oh.
Work on the handwriting. Okay. Question 4. We have an organism called Daphnia. It's a freshwater crustacean that can detect chemical cues produced by the predator Notonecta glauca. Interesting. Now, researchers raised the Daphnia clones and observed that those exposed to Notonecta chemical cues during development produced larger protective crests. The crest height and width are measured in ratios to body length. Okay. Now, and the genome does not change. The genome does not change, but the environment changing will affect them if they are developing in that environment. Do you guys get the idea? So if they're put in a container with this predator, they will become, they're gonna have larger protective crest. But if they grow in a container without this predator, they're gonna have shorter protective crests. That's the idea.
So like their body can tell if the predator is inside.
Yeah, their body can tell. Exactly. See, they do not develop large crest in the absence of the predator. Okay, and as we can see, if there is a predator, look at their crest height, it's more. Look at the crest width, it's also more to protect them, basically. Okay, A, describe the factors that influence an individual's phenotype.
Environmental pressures.
Environmental pressures.
Yeah, like danger.
That's not enough, though.
Wallah.
Oh, this has to do with unit 7 and 8, right?
That changed the expression of genes.
No 6, I think.
Chapter 9 and 10. That can affect gene expression. Yes, you must say that. Because if there is only environment without affecting gene expression, that's not going to affect anything.
Mister, is the chi-square thingy coming on the exam?
Oh, yeah.
That's so hard.
Can you skip it?
Skip it, you lose points. But I made a video about it.
So I have a question.
Yeah.
Actually,
I did.
no, wait. If you listen, if you do every single multiple choice but skip every FRQ, how much?
Why would someone do that? You're already in the test.
Huh?
Maximum 3.
Let's go.
That's if you get everything correct, like 100%. Why would someone just skip that for a Q? You're already in the exam.
See how hard they are. What is this?
They're not hard, just
I'm assuming someone who can get a full in the MCQ can probably master the FRQs. I'm
So you know how I
assuming.
am in
No.
MCQs.
Really?
No.
I'm saying how I am in MCQs. You
No, he
remember
doesn't.
Every
I
single,
actually don't.
Bro, I'll teach you
know,
every single lesson. I'm good at it.
what every single.
I want to see you in the exam when you're alone.
I've done two MCQs. Two or three.
Alright guys, so let's move on. Explain how the presence of Notonecta chemical cues affect gene expression in Daphnia. It causes expression of genes that lead to more crest height and crest width.
Mr. I don't get it. How did they link this to gene expression?
Well, if the genes are expressed and those genes can result in more crest height and more crest width, it's because there's a predator in the water. How can you get more crest height and crest width? There has to be a gene that has to be expressed or turned on, basically. Yep. C. As a
Can you see more expression of genes that are responsible for height?
for crest height and crest width?
Yeah.
Well, yeah, that's what I said, right? What did you say again? More expression of genes that result in what?
That are responsible for.
Yeah,
I have
exactly.
a question. For like when you finish your AP, can you like check the paper, see what you got wrong and what you got
I
right?
can't.
Doesn't it get posted?
No, but I heard you can buy your FRQ paper.
Voila.
Like you can buy FRQ answers paper. I
Not
heard
your
you
own.
can buy it from them.
Where'd you hear it?
Hmm?
Where'd you hear it?
From College Board.
Like I can.
No, like after the exam.
But why are they selling it though? Like,
After getting your mark.
After you get the mark, you can like request it to see it.
Mr. When do we get our marks?
July. So like if you get a 5, request it and put it like on a wall. Frame
We
it.
have to live in fear till July.
I know.
So they're like the government here. They will make everything money. What is this? It's my own paper.
Right?
I think like expensive too. Like
It's like more than $50.
yeah,
Yeah, that's not. I thought it was like.
you gotta pay for the mail as well.
Okay, rich boy.
It's a good business, honestly.
Listen, this is not fair. In America, you do an AP for free. You get your answers for free.
Really?
Yeah.
How do you know?
Do you live in America?
No, she's just
know.
guessing.
It's not for free, but it's less money.
She probably saw it on TikTok.
But it's less money. That's true.
Yeah, obviously.
I think like $30 instead of 150. I think.
Sir, I'm so stressed.
We'll do the blueprint. All right, guys. Part C.
I'm telling you guys, next week we're both studying together.
As a follow-up experiment, researchers placed the Daphnia that was exposed to the chemical in a tank without the chemical. Guys, part C. The Daphnia reproduced by a single parent asexually, and the offspring developed in the tank without the chemical cues. So, guys, if the offspring developed in the tank without the cues,
It will have the normal height.
it's going to have the same crest width and height as the control, which means not so much, right? The white ones. So it will have similar crest width and
So that
height.
one was easy. That part is
So I wrote that it will,
That's actually true.
it will have a normal crest width and height
Normal?
instead of control.
But sir, can you explain the question?
Normal means what? Okay, so look.
A control hamster.
Look, you are taking Daphnia, which is this organism, and you're placing them inside a container that doesn't have any chemical cues that cause them to develop the protective layer.
basically how it would be if it didn't have anything to affect it. Normal.
So they just grow normally,
You know.
like the control, you must say, like the control.
So the first one I understood so far. So.
I'm so good at FRQ.
I've never said that in my life.
Part,
You literally said
I've
that
never
to
said
me.
that. I said MCQs.
I swear you said that to
Wallah, I
me.
said it.
guys, we're all on the same team. Part D, provide reasoning to justify
I
your
swear
prediction
you
to
said
part
that.
C.
On my mom's life, you said that to me.
Okay, you know what? He said or not, guys, we're on the same team. Are we on the same team?
No.
Yeah.
Without being exposed to the chemical, the genes for more crest width and height won't be expressed. Okay, easy.
So, you know, Daniel told me to study for AP like practice from Gizmo, the app. Have you seen it?
No.
But sir, I'm studying from Khan Academy.
Guys, the blueprint. You know what's funny? Every year I put like a template for students to follow, and you know what they do? They always avoid it. I don't know why. I'm telling you the thing to do, you know? But you guys always go the other
So
ways.
you're telling me if I finish this...
I swear, I swear, okay, I swear.
Okay.
Yes, I'm telling you.
Like, you actually
But don't go to Khan and don't go to, I don't know what that is. What's
So
guess?
is it useless?
Gizmo?
But I don't think it's useless. Like I think it's
My friend, the blueprint. That's it. The blueprint. You cannot do both, okay?
You sound like you're advertising your like
we're
company.
gonna find.
I get nothing out of it. You guys already have the drive. Nothing, I swear.
بس خانة قاعدة يهمني يفيد.
Khan Academy is already for
I'll
free.
get a 6 for Mr. Not only 5.
Look, if I get a BISC student to get a 5 in my class, I think I'll get like
I
torn-
got you,
a lot
Mr.
of good reputation.
I got you, trust me.
Because BISC always
We
hard,
got you.
has a hard time getting those 5s.
Mister, these 3 students, they got you.
Alright.
She's aiming for a 3.
So guys, a student collected samples of cells from a woody plant. Okay. During the spring. And produced the drawings below. Okay, which one do you guys think has more mitosis?
The second one.
Right, how do you know?
So look at the stages.
Exactly correct. 1, 2, 3, 4, 5, 6, 7, 8.
So why are they like that? Why do they?
Because they're dividing. Versus only 2.
What?
So there's more mitosis in the root tip.
Yeah, I know.
Then why are you asking?
I'm asking because you're not in the right order.
Guys, they're gonna hear your arguments. I don't think they would like that.
Who is they? Mr. No's gonna watch.
Look,
this.
you'll listen to others' recordings. And they listen to your recordings.
I was listening to Dada's recording. The day was so weird.
Mr. Actually, how do you teach them? Mr.
I don't.
Quiet for 1 minute.
They do the talking. Okay, so guys, part A. Describe the role of mitosis in the growth of a plant.
To copy something like
To produce
More cells.
genetically
identical.
Cells.
identical
Cells.
cells.
Asexual cells, right?
It's doesn't matter for growth and repair.
Can't you just say to copy?
Man, you're making it too easy. No.
Mr. That's what I meant. Like, how am I gonna word them? This is how I would word it.
Yeah, over time you're gonna learn if you do the blueprint.
Yeah.
If you don't do other things just because someone says do them.
them.
Good.
Lice.
The app?
Yeah.
What app?
Hanukkah.
Oh, it's an app now? Okay, take it out. Explain why there are more cells observed in mitosis in root tip than woody stem.
Because that's where the division takes place.
The root tip exactly has more dividing cells. Wow. See? It's not that bad.
See, I'm also the left one.
Very good.
So I didn't understand the question.
The question says, why would you have more mitosis in the root tip than the woody stem? Because the root tip has more dividing cells and is growing more.
More lastly.
More mitosis means more growth.
Also, in Sharuto, I thought I would look down on the pattern.
Oh, okay. Part C. Refine the model and the space provided for your response by using the letters from the 4 of the cell labels of sample 2 to represent the sequential steps of mitosis.
Okay,
Which one is prophase?
the first one.
E.
Yeah.
E, right? The first one here. That's prophase. Which one is metaphase? That's like the easiest thing. D.
PMAT.
Yep. Which one is anaphase? They split up.
And then C.
Telophase means they go back
Two
to
different ones.
2 different cells.
Mr. This is so easy.
Yeah, wallah.
Wallah, that would never come up. trust me.
This is making me
Okay.
have higher hope.
Explain how the explain how the arrangement of cellular components during the step of mitosis by cell D, which is the metaphase, in sample 2 facilitates the proper distribution of the chromosomes to 2 daughter cells.
So we got this back in our bio quiz 6 times.
Yalla, go ahead then.
Spindle fibers and stuff, right?
The
So
sister,
they have to,
yep,
like, line
sister
up.
chromatids get pulled
Mr. The question is so easy, but it sounds Chinese when like you're
to opposite poles
I swear, wallah, wallah. Like the answer, it's
to ensure equal. Guys, please like.
Doesn't
Distribution.
that happen
Thank you.
in
But we've
meiosis?
done
I
this in
know,
the
I've
quiz
done
so
this.
many times.
Of chromosomes.
But this doesn't happen in mitosis.
It does.
It does.
Also in meiosis. Okay.
doesn't it happen in the interface?
No, it's an
Interface.
anaphase.
Oh.
Anaphase, right?
Don't watch the recording anymore. Anaphase
It
Yeah,
coats
yeah,
the
it's
parts.
an anaphase.
Yeah, she's right. She's right. Let me just make sure my.
They're all in the interface.
Are you
Mhm.
Aren't they, Mr.?
Interface is like
Content,
preparation for the
content,
cell.
Yeah.
Yeah, it's like
Bro, you're in a whole different thing.
Oh my god, never mind,
This is
never
the
mind,
beginning
never
of the
mind.
year.
Never mind.
You must say the equator here, by the way. You must say the equator.
Yeah, I said that. See,
Okay.
this one.
Lining up
Equatorial line. Equatorial plate.
on the equator.
and the equator.
What?
Yeah, we call
Okay.
it different in school.
Can I call it
Can get
equatorial,
pulled.
mister?
Equatorial.
Yeah.
Yeah,
Isn't
yeah.
that what we called it?
Equatorial plate.
Yeah.
Is that the name?
Yeah.
Good.
Basically that line in the middle.
All right. Now look, guys. If we look at surface area to volume ratio, which one here is the best? Which rectangular solid?
The 4. No, wait, at the 5.
Five.
The fifth one, exactly.
Mister, there's an equation for this, right? It will come back.
SA
Yes.
over
So
V.
easy.
Oh,
Yes.
No, SA
There's a
equals
formula
to
sheet. We don't need to memorize.
mass
it.
over volume.
No, it's.
No,
pi
that's
square-
actually density.
No, it was like pi square something.
Bro, no.
R squared? Okay.
That's for
Guys, let's move on.
that's for volume of triangle or something.
Guys.
Is the formulas going to be given for us?
Yes, yes, yes.
Oh, it's in-
A. Identify the rectangular solid that represents the cell with the smallest amount of plasma membrane per volume.
So what does this mean? It's just
It means the least surface area to volume ratio. That's what it
the
means.
first one.
Yep.
Mister, this is what I mean. Like the questions, they write them in such a hard way, but they're actually easy.
Because it takes time, you will get used to it.
So I'll do
See me in a week, I think I'll see a better version of you because you would have started the blueprint.
Yeah. Trust me, I will not let you down.
Guys, should we go Sunday and start it together?
That's what I've been saying, bro.
Just go somewhere.
Mister, I've been telling them let's go out and study together.
And?
They're not listening.
But for me,
said yes.
Okay.
We all said yes.
Guys, part B. Describe the relationship between the dimensions of the rectangular solids and their surface area to volume ratio.
I know this one, Mister. The bigger the rectangle,
No,
the
no. As the length becomes more,
With increasing
as the height becomes less, as the width becomes more, the ratio will become more.
higher.
Yep. So the ratio becomes more with more length, more width, less height. C. Using the data from the model, evaluate the hypothesis that the cells shaped like a rectangular solid 1 and 2 are better suited for slow metabolism and long storage of energy compared to 4 and 5. You must look at the
Surface area.
to volume ratio. No, it's the ratio, the whole ratio thing, right? So 1 and 2 have low surface area to
It's
volume
like
ratio.
It's small inside, so like they get around quicker and stuff, right?
No, بالعكس. Look, if the ratio is smaller, you're going to have slower metabolism, which means now you can save more energy because you have less efficiency of things. Yeah.
So the less, the better.
No, the less the worse.
Oh.
But for this question, they're asking for worse.
Oh.
Yeah. So we're going to say that for C, solids 1 and 2 have a smaller surface area to volume ratio, so they will have less efficient metabolism compared to solids 4 and 5.
So, Mister, if they have smaller surface area, it's gonna be less efficient.
Exactly correct.
And what if they have more surface area?
That's better. Yep. The opposite. Okay. D. Based on the trends shown in the data, explain why some rectangular cells include folds or projections in the plasma membrane. This is to increase the surface area to volume.
Villi?
Villi, yeah. Whenever you see villi or fold or projection, it's about increasing that surface area to volume ratio.
So
So
that
it's
basically
easy.
means
Sorry?
increase.
Oh, we're finished?
That means increase by different words, right?
No.
بس explain بس الجد.
No, no,
Yeah, you're right. Thank you.
Explaining بس الجد.
Say
Yeah.
it.
I swear.